it's really been a journey over the last decade, with the identification of mutations that lead to the development of myelodysplastic syndromes. And this has really been like, we're still like at the surface where we try to see if we can develop targeted therapies for those mutations, similar to what has been done in AML, where we have FLT3 inhibitors, IDH inhibitors that really have provided new treatment options for patients with those dedicated mutations. However, in MDS, those mutations are not as common as in AML. And right now, none of those medications, neither FLT3 inhibitors nor IDH inhibitors, have actually been approved by the FDA for treatment of MDS. Sometimes we use those off-label, but there's not been a dedicated registrational trial of those agents. However, this is really kind of provides the paradigm for where we try to develop novel targeted therapies based on a more granular understanding of the molecular landscape. For example, there's like splicing mutations are among the most common recurrent mutations in patients with myelodysplastic syndrome. And there are have been very difficult to target. There have been trials that have been done, and there was an early signal, a few patients with hematologic improvement, but overall, that particular compound didn't move forward, at least at this point. And we have also led a trial that's currently that's about to be published in leukemia with an RBM39 degrader, which is a key factor in splicing regulation. And we'll also have that with both AML and MDS patients, also has limited therapeutic efficacy. So I think splicing modulation would be a very interesting target, but we still need probably both better drugs to target this, as well as maybe still more advanced understanding of the molecular landscape. And the other part that's been ongoing are like, can we target TP53 mutations? For example, we've seen APR-246 in combination with azacitidine. There was a randomized trial where we still only have the press release as far as I know, but that did not meet its primary endpoint. Similar to azacitidine and the anti-CD47 antibody magrolimab. That was also something that had very promising data in a phase 1 trial, but then the phase 3 trial has also been reportedly negative. We've always also tried to harness the immune system with checkpoint inhibitors. They're still ongoing trials, and we'll see what they have to, what they'll eventually lead to. But I think there's been some progress certainly in the understanding the molecular landscape of MDS. However, this is not really translated yet into clinical practice. So hopefully in the next few years, we'll make some progress there as well.